Okay, well, thanks for the kind introduction. Thanks for dedicating your late Friday afternoon to this. I don't know what they did to compel you to come on that Friday afternoon, but here I am, and I'm grateful that you're here with us, and I look forward to the discussion. Let's see, is this going to transmit from here? I switched here, but it hasn't gone there yet. There we go. Okay. So the talk that I prepared is kind of a mix between talking about kind of how to do kind of innovation and entrepreneuring the way we practice it, and then give you some examples of the kind of projects that we've been working on. I'll also touch on the Moderna experience because I think it's instructive for all the rest of what we do. So I titled this Pioneering and Entrepreneuring in the Age of Biology. And before I even start, let me just kind of say what we mean by pioneering and what we mean by entrepreneuring. So pioneering isn't a very typical word. You hear a lot about pioneers, but you don't actually think about the activity involved in pioneering. And I'm going to try to describe to you how we think about that, because we think that you can be thoughtful and planful and consistent and systematic about doing this type of scientific leap taking. And that's what I mean by pioneering. Pioneering we use in the version that describes, for example, pioneering species. These are the species that first colonize an uninhabitable space. If you look it up in the dictionary, that's how we think about pioneering. It's really to colonize new opportunity spaces, new solution spaces. Then entrepreneuring. You probably have never heard that word before. And let me explain why I at least prefer to use this word. I've been involved in one or another form of life as an entrepreneur for 36 years. Since I graduated from MIT. And the interesting thing is, over time, I started realizing that the word entrepreneurship left me a little bit unsatisfied, because I had to look it up. Well, first, I'm just trying to break the ice a little. It's a Friday afternoon. You might know where the word comes from. I don't know if you know where the word entrepreneur comes from. It comes from a French word, and the word is entreprendre, which means to undertake. So the literal definition of entrepreneur is undertaker. And if you've worked around startups, you know that there's quite a bit of that you have to engage with. But that's the funny part. The more serious part is the ending of the word, which I have a problem with. So words in English that end in ship usually mean the state of being something. So leadership, craftsmanship, sportsmanship, friendship is the state of being something. So when you say entrepreneurship, it's the state of being an entrepreneur. Now, I studied engineering. I don't do engineership. So what's the difference? The ending of the word ing means actually doing something, not just being something. And I would argue that many people, entrepreneurs in this room, don't just think that when they wake up they're doing entrepreneurship. It's during the day that they do entrepreneurship by taking a set of steps to getting better at it. So I'll just lay out that, when we use the word entrepreneuring, we mean by both of these things actual activities that you can get better at and be professional at. So this will carry through much of what I'm going to describe today. So I'll also kind of ask you a question to keep in mind during this talk, and then maybe in the dialogue at the end we can revisit it. And the question I'd ask you to kind of think about is, why do we collectively expect extraordinary results, extraordinary outcomes from reasonable people doing reasonable things? And before you say, we don't, surely we don't, if you ask yourself as an investor, what do you look for? You do due diligence, because you want people to tell you just how reasonable an idea is and just how reasonable people are. When you give grants, you're assessing how reasonable the proposition is, right? Who in the world would give a grant to a completely unreasonable person doing an unreasonable thing? And so by that logic, we should expect pretty ordinary outcomes. And in fact, when 100 people are working on the same thing, then for sure we should expect very ordinary outcomes, because it'll all be diluted a little to this, a little to that. So I would argue, and the central thesis of what I'm going to try to show you and convince you today, is that if you want extraordinary outcomes, you have to find unreasonable people doing unreasonable things and hope and help them get to a point where what they do eventually becomes reasonable. And that's the path that I'm going to try to describe to you today. So now we operate within Flagship in squarely in what is otherwise a pretty interesting environment, which is kind of broadly what I'll call applied biology. In the last century, applied physics, applied chemistry were very much what drove a lot of value creation, a lot of new solutions in energy, in materials, in lots of things. Applied biology, actually I had to look up if anybody ever uses these words. Very few people do. I kind of fooled you if you had thought you had heard of it before, because applied biology hasn't really been a thing. But actually now, I would argue, in this century, we're kind of enjoying a moment of applied biology, applied to many, many different things, not just therapeutics, and I'll come back to that. And interestingly, any time a discipline, a scientific discipline, starts becoming engineerable, the resulting value usually goes up dramatically. That's been our experience in the past, and I think that's going to be our experience with biology. Now, the kind of things that applied biology is extending into, as I said, is not just biotech products, it's not just drugs. We saw what happened during COVID-19 in terms of both the diagnostics, therapeutics, and the vaccine solutions that came out of essentially applied biology. We can go to food security, where there are many, many needs that are being tackled. Cancer as a big, big plague. Broadly, climate change. There's a lot of things that are going to be requiring a level of combination of technology and biology. Much of the solutions in agriculture, much of the solutions in renewable fuels, all are benefiting from this interface. So I would argue that while 40 years ago, when I came down to MIT, people were talking about biotechnology impacting energy and many other things, I think 40 years later, we're at the beginning of when that's going to be possible. And so you're going to hear a little bit about that as well. Let me broadly describe what the organization that I have the good fortune to run does. The Flagship operates as a company that conceives, creates, and resources first of their kind companies. That's what we mean by pioneering. And they're not just companies per se, they're actually companies that are based on bioplatforms. Every single company that you're going to hear about today has at its core a bioplatform. And we're applying these bioplatform innovations to transform human health or the planet's health. And by that we mean sustainability. We were founded in 2000. We have been involved in the founding of over 100 companies. 30 of those have gone public. The current ecosystem of companies that operates today in 2023 has about 60 different clinical programs and 150 preclinical programs across it. Flagship itself really has two levels of operation. I'm going to describe this to you today very briefly. One is the core Flagship unit, that's about 350 people. And then the broader enterprise of Flagship, which is all the entities that we own and operate before we spin them out, that's another 750 people. So altogether, the organization is a little over 1,000, the vast majority of which are scientists and MDs and engineers. About 100 of those are dedicated to just do the kind of exploratory breakthrough innovation that I'm going to go through a little bit of detail for you, just to give you a sense of scale. We file at the central Flagship labs some 400 to 500 patents a year, spanning many, many different areas of activity. And our capital comes from a series of funds that, in that one regard, we look like a venture or private equity firm, but all of our funds are directed to companies that we create and own and operate. So in that regard, we don't actually have quite an investment mindset, rather an entrepreneur with capital and expertise mindset. Over the last 20 years, what we do has shifted somewhat. In the first 7 years, we focused a lot on how to create an institutional way to do venture creation. So where it's not about an individual with a business plan, but you can actually, as a team, conceive and launch companies centrally. This is now a long, long time ago. This has become more popular these days with the proliferation of venture studios and incubators and many other venture approaches. We started there quite a while ago. And then we also were looking at academic science as the mainstay of how we got access to innovations. About 2007, 2008, we started working more and more on internally generated platforms, internally generated science and IP, and still continued to improve and started thinking about this entrepreneurial concept as an institutional act. Since 2017, we've been doing both pioneering and entrepreneuring at some scale, and you're going to see that in the next couple of slides. But in each generation, there's companies that have come about that are developing. So you've heard me talk about this before. There's three things that we think drive value within our organization. Pioneering, which is where the science comes from. Entrepreneuring, which is where that science converts into value. And scaling, which is what causes us to be able to grow that value and capture that value. And we think of each of these things organizationally very carefully. So there's a couple of things that I'm going to tell you, again, at a high level about Flagship, just to provoke some thoughts. One is where we innovate, and I'll try to describe to you that we try to innovate beyond adjacencies. That's the only way we're going to be able to find new pools of value. I mentioned that the bioplatform concept is central to everything we do. And by the way, if you're wondering what do we mean by platform, we don't mean technology per se. We don't mean sequencing or other approaches. What we mean by bioplatform is a common new capability. It could be a new molecular class, it could be a new finding in biology, or a new technology, but that leads to many, many products that can come out of that same core advance. So by platform, we mean something that if you invest enough into and can possess, can generate many, many products, not one-off products. That's what we mean by platforms. All right, so let me dive into this. So let me explain kind of this idea of where we innovate. This is a very, very simplistic depiction of the history of innovation, or the let's say, totality of innovation. Very simplistic, at least according to me, because it's Friday afternoon. I can't do this anymore than drawing circles. In gray, I'm trying to depict the current solution space in any given space. This could be immuno-oncology, this could be virus, the virology, it could be renewable energy. In any given space, if you take everything that's known and everything that exists and draw a circle around it, you could argue that, okay, that's one representation. And then if you ask, where do people innovate? You'd find that most incumbents innovate either inside that circle, and that's called niche innovations, filling opportunities that aren't yet taken, because they have a huge advantage because they own the space, or just outside that space. And that's the space of adjacencies that you see those little gray dots in. But beyond that, there's a whole lot of other people who innovate in that zone of adjacency, and that is where a lot of startup innovation happens, a lot of academic innovation happens, because that's where the next opportunity lies. And so adjacencies, which I would define to you as things that are reasonably far from where the edge of knowledge is, but not unreasonably far, such that you can apply due diligence skills and risk-reward analyses. That's what I mean loosely as this zone of adjacency. And there's two reasons why people invest a lot there. One, most experts will guide you there, because most experts are not going to tell you to work on something that's 5 leaps from where you are today. That's what makes them experts, is the authoritative knowledge of what will work next. And so that's where they'll guide you. The second reason is because if you get something in that blue zone, somebody will buy it from you very often. And so it's a pretty good place to be if you're trying to actually create return on the risk that's being taken. And so that's where I would argue a lot of value is created, and certainly at large scales, it's done a very good job all around the world. The question that we, over the years, started asking ourselves is, could we consistently operate outside of this zone? And that's what I'm going to try to describe to you to make your Friday afternoon a little exciting. And so the question is, why would you even operate outside of this zone? Well, one thing is there's a lot less competition there, right? Most people are not going to be able to go. We're describing zone now beyond adjacency where any expert will tell you it won't work. In fact, every expert will tell you it won't work. So that's just if you want to get a sense where you can't point to scientific papers that say, okay, this has already been done, and I'm going to take this next. So this is kind of in the middle of nowhere. It's white space. Now, you might go there because you might unravel and find new value pools. You might find completely things that have never been done, and you can create a very interesting new way to create returns. You don't want to go there because you have no idea where value is. There's nobody to copy. There's nobody to leverage. Nobody can tell you you're doing the right thing, let alone telling your investors. So it's a pretty scary place to operate. Now, let me explain one thing. It's a little bit, kind of tedious, but just bear with me. And then after that, I'll get into more scientific stuff. This is kind of what I understood after 36 years of doing this that I wish somebody had told me a long time ago, because it now drives a lot of how we think. So what I've tried to depict here on the left side is kind of on the y-axis is risk, right? And what you see is kind of low risk opportunities in the current and then higher risk opportunities in this adjacency zone, right? And you can see again depicted the circle thing in a different format where the further you get from the current, the more risk there's going to be important. So you kind of diversify your bets. Now, the advantage of operating there is that you have relatively low uncertainty. In other words, you can make risk assessments. By the way, let me explain what I mean by risk and uncertainty. The way I'm using it, the way economists use it, I'm no economist, but I've been told, is that risk is about known or estimable probabilities. Uncertainty is about unknown or unestimable probabilities. So we typically use these words interchangeably, but let's just for a moment say let's not use them interchangeably. So if what you're proposing to do, there's no basis to know what's the probability of success, that's uncertainty. That's not risk, right? And vice versa, if you're proposing the 10th version of a whatever, autologous cell therapy approach, then you could say, okay, this one has this advantage and it's going to lower the risk at this stage by this much, or if I'm going to do biomarker-based patient selection, I'm going to improve my odds. That's more in the risk space, right? So the danger, I would argue, in adjacency-based innovation, however, is that it's pretty wide open for everybody to walk in. You have no idea if you're competing against startups in different places of the world or large companies who are secretly working on something very similar. So that's kind of what you have to work on. And I would argue, just as again, as an original thought maybe, that counterintuitively, chances that there's low risk opportunities left in adjacencies is pretty low. Because if there were, I think you should assume other people would have taken them, right? Because it's definitely not an empty zone of work, right? Now, what about to the right of this? And this is kind of what I'm calling kind of the future. So working on things that are going to be possible maybe in the future is inherently uncertain, right? And I would argue that it's really a good thing to avoid because you can't know which is going to work, which is not going to work. But actually, ironically, this is my kind of a bit heretical suggestion to you. I think if you accept that risk and uncertainty are somewhat different things, then how you should prosecute uncertainty could be quite different than how you should prosecute risk. And importantly, if you open up a space which is uncertain and you show how something can be done, then there's no reason to believe there's no low-risk things to do, right? So think of it this way. You go to a place where everybody has access to, and the chances are there's no low-hanging fruit. We use that expression a lot, low-hanging fruit, low-hanging fruit. Well, low-hanging fruit doesn't exist in places that other people live. But if you go to some place that no human has ever been on, or no animal's been on, what's the chance there's no low-hanging fruit? Well, I can't tell you what the chance is, but I can tell you that nobody's picked it already. So I would argue, and our experience has shown, that if you are willing to embrace uncertainty, which investors are extremely uncomfortable with because they need some basis to go on, understandably, but if you're willing to do that, and if you can kind of get that uncertainty reduced, how do you do that? You generate scientific proof. You actually reduce to practice the thing that you're claiming you can do. You kind of fill it with a lot of invention so you can protect the whole space as opposed to one tiny little innovation. You build multi-product platforms, so you have many different options to pursue as opposed to locking. All these things are things you could do to try to tame uncertainty. My argument would be, for the rest of this presentation, that you might actually create lower risk, albeit initially uncertain opportunities, than what you could in adjacencies. That's at least our belief system. So you might say, okay, whether you believe it or not, how do you do that, right? So let me explain kind of how we operate within Flagship. So there's about 100 people, I said, who do this kind of pioneering innovations. Well, this is the process we follow. We're actually relatively boring. Most people would think that entrepreneurs and scientists want the freedom to create and to do all sorts of things. Well, the reality is we follow a process. And we convinced ourselves 20 years ago that if we were going to do a crazy thing and go to the edges of the edges, we should do it in a consistent way. Because I believe you can't learn if you do new things in a new way. You got to hold one of these things constant. If you do things kind of sort of in this new way, in the same way, and you keep changing the output, at least you can learn. But if you just wing it, improvise, and you're trying to create totally new things, and it's really hard to know what's working, what's not. So just keep that in mind as I describe this process. Pretty straightforward. By the way, I hope I'm saying some of these things it probably sounds totally kind of amorphous, but hopefully it'll make sense as I give you examples. So the first step that we use to do this kind of activity is what we call leaps of faith. Now, I bet you didn't think Friday afternoon in a science-oriented talk, which I'll get to in a minute, that I'm going to talk about faith, right? So I don't know how many of you have. I hadn't until about a year and a half ago. I actually had to give a talk, of all places in the Vatican, during COVID. And they wanted to know about the interface between science and what we did in vaccines. And it was interesting because I realized that I never really thought about this word that they, of course, often use, which is faith. So faith means belief without facts. Faith is not just a religious concept. It's just an English word. It means belief without facts. And of course, in science, we are taught not to believe things for which there are no facts. That's the antithesis of science, right? But here's the thing that I would at least ask you to open your mind to. If you believe in something without facts, but then go get the facts to support what you believe, at the end, if you have the facts to support what you believed without facts in the beginning, what is that? Science or not science? I would contend that's science. In the end, if you have facts that support what you believed without facts, you have made a breakthrough. You have basically leapt to something that nobody believed, but now you have facts. Until that time, what are you doing? You're acting on faith. And of course, most scientists would never say that, right? I'm 60 years old now. I've done this for 36 years. I will say that. There's nothing wrong with actually imagining solutions as a scientist, as an engineer, believing in them, even though you don't have data, long enough until you convince yourself that it's worth the journey, get resources to get on that journey. Don't forget to go get facts, because a lot of times people forget that and they just go implement it as though it's going to work, and then they find out later there's nothing real. Don't do that. But if you go get the facts after the fact, I think that's another way of doing science. Now, that's not the way we do science or innovation traditionally, and that's not the way you're going to get a lot of grant money to do that, because people don't like what I just said. People want to believe that every incremental step is based on every other incremental step. I like challenging people. Imagine you came up with some completely new, novel idea. And imagine if you went to a lab and generated some data, and you actually showed that you can actually do the thing you came up with, even though it had nothing to do with what had been done before. Now you go to publish it, right? Imagine submitting a paper to Nature or Science or Cell that has no reference section because there's nothing that you based it on, right? You'd be laughed out of the room. You can't get published. So what do? You add 20, 30 references, even though it's not at all what you might be doing, because it's inconceivable that you can actually leap. So I'm just inviting you once in a while, and I'm sure all of you do it, by the way, secretly. It's just that we don't talk about it, that it's okay to just say, you know what, let me leap temporarily and see if there's any value outside of what the obvious next steps are. So that's what we do. We call these what-if kind of explorations. Now, you might say, well, how do you know which way to go? And so one is, we don't do it once, we do it about 100 times a year, different directions. But then the other thing we've learned over many, many thousands of these that we've done over the years is that where you land, this black dot that I'm showing you here, the picture is not coming out too well, that is by definition a bad idea, right? So imagine what I'm saying is what we do for a living is coming up with bad ideas. That's what we do. And a lot of people tell us, what kind of people do you hire that can come up with these brilliant ideas? And I disappoint myself, we don't have any brilliant people that come up with great ideas. We don't. And I really mean this, by the way. I'm not being lighthearted. See, and this was, by the way, we published the basis for this way of innovating a couple of years ago at Harvard Business Review, together with Professor Pisano from Harvard. So here's the second part, which will be all dear to all of your hearts who do work on biology. See, it turns out that the initial idea that causes you to jump someplace isn't what's important. What's important is the descendants of that idea. And so this notion of descendancy that we all have learned based on Darwin's pioneering work is actually alive and well when it comes to ideas. So you don't have to have a great idea. You just have to have an idea that you subject to variation and selection in the first instance, long enough to come up with variants and variants and variants of it that actually start looking like, well, you know what, if this could be done, it actually could be pretty valuable. And so your goal is not to get the black dot to be this red dot on the bottom. You just want to run a process that will cause it to emerge. So we kind of call this emergent discovery. Now, I'll give some examples of this because I don't think this will sound too abstract, but take my word for it. Spending a lot of time coming up with brilliant fantasies is not a good use of time. In this day and age where ChatGPT will give you hallucinations for a dime a dozen. By the way, if I were you, if you want to come up with interesting leaps, use ChatGPT, because it has no idea what it's leaping to, but you might be able to look at 10 different things that it says and actually find in there an idea you might not have come up with. So long as you're willing to iterate, and gather facts to see what is real and what's not real. So this notion of descendancy, kind of a Darwinian process this is what our team practices a lot. This is what we do for a living. And so for example, let me give you an example. We might say, what if there are proteins that are made in our bodies that we didn't know existed, right? So you might say, well, what is that? Like, why do you say that? And so, for no good reason, by the way. It's just a what if. It's like a black dot. So then you can go to people, experts in genomics and proteomics and cancer and whatever, and say, we think there's proteins we have no idea exist. What do you think? Literally, that's the first thing we'll do. We'll go to experts. And then they'll say, well, what do you mean? There's 22,000 proteins. We knew that since the genome was done. Maybe it's a little less. And so you go, okay, but what about other things, like not coming from canonical RNA translation? So if you say that in a serious way to scientists, they'll dismiss you right out of hand, because you need proof to be able to engage them. But if you do it in a lighthearted way, say, well, like, what if it's possible? Like, where would they be? And then you start thinking, well, wait a minute. There's a lot of mass spec traces, by the way, that cannot be tied to RNA kind of transcript data. It's been well known. You cannot actually trace back to a known exon a lot of protein sequences that you get out of mass spec. So what is that? So people will say, yeah, but that's junk and it's little things and this and that. I'll roll the clock forward. Several iterations of this later, because people told us, well, how's it being made? Or where does it come from? Where's the DNA coming from? And we said, non-coding DNA, or non-coding RNA. And they said, well, how can non-coding RNA become proteins? So we had to go back and come up with a way to propose that non-coding RNA could come up with proteins. And if you do this kind of in a kind of idea of space first, it turns out that you will come up with a version of this that you can go test. Now, you might be sitting here thinking, what is this taking us? We did this 3 years ago, and about 2.5 years ago, we started going into labs and looking for non-coding sequences that you can show the corresponding protein to. And you may have read in the literature, because now it's beginning to appear from academic papers, there are tens of thousands of peptide, small protein and peptide sequences that are expressed through non-canonical mRNA sequences in human cells. They are differentially expressed in disease versus not. They are differentially expressed in different cell types. And none of us, including not us, have any idea what these things are doing. But to the extent that we think we know the human proteome, and therefore we can bound the problem with what are these 6 cytokines that are involved in this pathway, there's a whole continent missing. Now, I'm using this as an example. You might immediately say, well, what are you going to do with it? How are you going to find proteins? How are you going to make drugs? All good questions. But what I'm trying to describe to you is kind of a bit of the descendancy of what is a what-if projection to a more actionable, let's go in the lab and see if we can actually take specific sequences and find them in human cells and correlate them with disease and not, etc. All right. The last piece of this is, what do you do if you find some place at the end, this red dot on the bottom, that you now feel like, boy, if I could just get there, that's a big deal. And the answer is, now the world of current literature looks very different. We call this reverse planning. If you know what your destination is, the way you read literature is different, because the things that matter to you have a lot more to do with where you want to get to than where you are. So keep one thing in mind. You could do innovation present forward, or you could do it future backward. Future backward is perilous in that if your destination is a faulty destination, you might be wasting a lot of time. But if you do this enough times and you're willing to iterate, at least what we would argue is you'll come up with different types of innovations. I'm going to skip a little ahead. So using this approach to pioneering and an approach that I'm skipping to how we create companies, this is kind of the world that we live in today. These are the companies that have come out, all of them, out of this process of exploration, followed by converting these into a prototyping phase, these protocols, converting them to newcos, and then to growth stage companies. So what you're seeing here is the current ecosystem. It's about 8,000 or 9,000 people who work in Boston and Cambridge. Everything we do is there, with one exception in California. So yeah, this is largely to just show kind of what exists today. But I'd rather get on to a couple of kind of specific examples of new science that we're working on. Important one thing I want to mention is a kind of a big conviction we have about platforms and modalities. I know that in the biotech space, there's kind of a pendulum swings between you need to have a product in the clinic that magically works, even though you have a 1 in 20 chance of it working, but your one is going to work, and versus platforms that have no products, which means that you develop the platform, but then there's no way to monetize it. Product platform combinations are a particularly interesting thing for us. And what I'm showing you here is in the gray below this yellow box, that my screen is being transformed a little in color, but is a bunch of platforms that have begun to appear of late and new modalities superimposed on the traditional small molecule protein modalities. And above in this box in the top are platforms that we have worked on creating the first or the second, one of the very first efforts in. We think it's the first in each case. For example, in Series' case, you might know this was the first company that went after making an oral microbe-based product to treat Clostridium difficile, recurrent Clostridium difficile. They just got FDA approval of the very first such product 2 days ago. In fact, when we landed yesterday, when we landed in Singapore at 5:30 in the morning, in Boston time, 5:30 in the morning, 5:30 in the afternoon, the FDA gave approval. That journey to go from knowing sequences of microbes in the gut to looking at recurring C. diff, to looking at fecal transplants and all the ways in which people were doing it, to saying there's got to be a way with a pill that you could do what people are doing with enemas and fecal transplants, to getting a product approved, kind of enables a whole modality, because now, in many other ways that actually your gut is dysbiosed, we think we can reset it. And so we're going after a whole bunch of other indications.